Mars here. I have promised one student that I would do a audio this week. So I have written some PowerPoints here, which you can read if you prefer reading, but for those of you who like to listen, I've written the PowerPoints so that I can just read them. So here we go. This is another one of those experiments, so I'd love some feedback from all of you on this. So first of all, let's do our summary so far. So this is a summary of, I guess, last week's content from the study guide and from the PowerPoints and all the other readings and things that you did. This is just a little short summary of the lifespan, the science of lifespan, I guess. So the summary so far: the study of lifespan development is scientific in approach, interdisciplinary in application, and concerned with the transformations and consistencies that occur over the course of human life from conception to death. So researchers in this area typically focus on one main area of development, so either physical, cognitive, emotional, or social. There are three basic issues of development that lifespan theorists and researchers address: the extent to which development is continuous or discontinuous, which is called stage-based, whether there is one general sequence of development or multiple paths, and the relationship between nature and nurture in human development. So let's get that again. So there are three basic ways that lifespan theorists think about life, and that's the extent to which development is continuous or discontinuous. So this is, do we develop in stages, or is it this kind of trajectory beginning down here when we're kind of born in a straight sort of line to old age? And whether there is one general sequence of development or whether there are multiple paths of change, more like a Mandela, I guess. And then they also look at the relationship between nature and nurture, which we saw when we looked at the history of lifestyle theories in week 2. Okay. So first of all, let's look at biological development. So biological or physical development is determined by genetically guided maturation, environmental resources such as nutrition, sunlight, toxins, accidents, illness and disease, and lifestyle patterns such as eating, sleeping, exercise and use of drugs. Physical development emphasises how the brain, nervous system, skeletal system and sensory system affect behaviour. And then we also have genetic foundations. So genetic foundations, we start life as a single cell. The first cell is a zygote. The zygote is created when a male reproductive cell, a sperm, combines with a female reproductive cell, an ovum. This reminds me of sixth form science. The male and female reproductive cells are called gametes. Gametes contain huge amounts of genetic information, and when two gametes fuse, they combine their genetic instructions, over 2 billion chemically coded messages, to form the beginnings of human life. So we know that after that zygote is formed, a thing called a blastoplast connects to the womb, and that's when our physical development begins. So the genetic code. At the moment of conception, humans receive between 20 to 25,000 genes. Genes are the basic units of genetic information. They are comprised of sequences of DNA, deoxyribonucleic acid molecules. Genes are arranged in a specific order and location along rod-like structures called chromosomes. There are 46 chromosomes that are organised into 23 matching pairs, except for the XY pair in males. One member of the pair is inherited from the mother and the other from the father. The pair members correspond with each other in size, shape, and genetic functions. Sorry, it just flipped to the next page. I don't know why. There are 46 chromosomes that are organised in 23 matching pairs, except for the XY pair in males. One member of the pair is inherited from the mother, the other from the father. The pair members correspond with each other in size, shape, and genetic functions. The 23 pairs of chromosomes and the new zygote contain the genetic instructions that will guide cell activity throughout life. Through a process called mitosis, chromosomes replicate themselves. This allows the zygote to develop into a complex human being. Each new cell body contains the same 46 chromosomes and identical genetic information. The nature and function of every cell in the body is determined by the precise location of genes on a pair of chromosomes, on the chain of chromosomes. For example, genes determine which cells will become part of the heart and how quickly the heart will beat. It's interesting, isn't it? I've got a really big heart and I don't know where that comes from in my family. But every time I go to the doctors, they're like, oh, you've got something wrong with you because you've got a really big heart. But actually I haven't. I just have a very big heart. And of course, that has some implications for me in terms of my fitness and my ability to exercise, my resting heart rate and all those sorts of things. So actually having a big heart, like Phar Lap, has actually been quite good for me in my life in many ways. And that's something that was determined by my genes, not by anything, lifestyle theories or anything that I've done since. So as you're going along, I'd like you to think about these things in terms of yourself as well. What kind of genetic influences have there been on your life? Are there some positives and negatives that you can think about? Are there people in your family? That sort of thing. So the vast diversity of human beings is explained by the nature of the processes underlying the cell division of gametes. Gametes are formed through a cell division process called meiosis. Each gamete has only 23 chromosomes, and in contrast to the 46 in the rest of the parent cell body. The selection of chromosomes to form the gamete's cell nucleus is a random process, so consequently there are 223, or about 8 million different chromosomal combinations possible. The ultimate outcome is trillions of possible genetic possibilities, establishing the sex of a child. Recall that chromosomes are organised into 23 matching pairs. In 22 of these pairs, each chromosome is similar to the other member. These pairs are called autosomes, with the exception of the 23rd pair, the sex chromosome. In females, this pair is identified as XX, in males, this is identified as XY. And of course, in intersex people, you will have combinations of X and Y, sometimes XXY, etc. The X is larger than the Y chromosome and carries more genetic material. The gametes that form in females all carry an X chromosome. When gametes form in males, the X and Y chromosomes separate into separate sperm cells. The sex of a child is determined by whether an X or Y-bearing sperm fertilises the ovum. Therefore, the father's sperm determines the sex of a child. This fact has led to the development of techniques that will allow parents to specify the sex of their child, which raises practical and ethical issues, which I'm sure you've all heard of, are aware of, like the whole kind of issue about genetic manipulation. So the next slide is about multiple births. So multiple births occur for two reasons. Sometimes a cluster of cells in the ovum splits off within the first 2 weeks after fertilisation. This results in two genetically identical zygotes. They are called monozygotic because they have the same genetic makeup. Differences between monozygotic twins can only therefore be attributed to environmental influences. So I'm going to show my age here. I went to the Proclaimers concert a few weeks ago, and they are musicians, and they are identical. But when you see them on stage, one is actually bigger than the other, and I was like, oh, they can't be identical because one's bigger than the other, but they are identical twins. So that's a very good example of how genetics are only part of the influences. There are also environmental factors. So a more common type of multiple birth results from two separate ova being fertilised by two separate sperm at approximately the same time. Twins produced by this process are known as dizygotic. They are more genetically similar than two siblings born at different times, however. Other types of multiple births include triplets, quadruplets, and quintuplets. The chances of having multiple births are slim, but increase considerably as women get older. And when you use fertility drugs. Patterns of genetic inheritance. Patterns of genetic inheritance are produced by the interactions of parents' genes. This interaction determines our characteristic. Sorry, everybody. For some reason, it's just, for those of you who are listening and not watching and didn't see that, it's disobeying me. Patterns of genetic inheritance are produced by the interactions of the parents' genes. This interaction determines other characteristics and also our disabilities and diseases. Remember that all chromosomes come in corresponding pairs except for the XY pair in males. Two forms of genes, one inherited from the mother and the other from the father, are located in the same place on the chromosome. Each form of gene is called an allele. If alleles from the parents are similar, the child will be homozygous for the trait in question. If the alleles are different, a child will be heterozygous for the trait in question. In this instance, the trait will be determined by the relationship between the alleles. Dominant and recessive inheritance. In many heterozygous pairings, okay, hang on, I'm going to stop this and do it differently. In many heterozygous pairings, dominant recessive inheritance occurs when one allele, or the dominant allele, affects the child's characteristics. The other allele, which has no effect, is called the recessive. For example, the allele for brown eyes is dominant and the allele for blue eyes is recessive. We can see this process at work by looking at the transmutation of phenylketonuria. PKU is one of the most frequently occurring recessive disorders. It affects the way the body breaks down phenylalanine contained in milk and other foods. If left untreated, PKU allows phenylalanine to build up to toxic levels, causing brain damage. Codominance and incomplete dominance. Not all single gene pair traits conform to dominant recessive inheritance. Sometimes each member of a gene pair is equally dominant or codominant. Inherited patterns can differ depending on the baby's sex. Recessive disordered cancers carried on the autosomes have equal chance of affecting males and females. However, the number of recessive genes called X-linked genes are only located on, However, a number of recessive genes called X-linked genes are only located on the X chromosome. Remembering that the 23rd pair of chromosomes in females is always a double X pair, while for males it is an XY pair. Genetic imprinting. Most human characteristics follow either dominant, recessive, or incomplete dominance patterns of inheritance. Geneticists have identified some exceptions. In genetic imprinting, alleles are imprinted or marked in a way that allows one member of the chromosomal pair to be activated regardless of its makeup. For example, diabetes is more likely to be inherited from the mother, from the father, asthma and autism from the mother. Genetic mutation. Harmful genes are created by a sudden change in a segment of DNA. This channel, known as mutation, can occur spontaneously or through exposure to hazardous agents. In germline mutation, changes take place in the cells that give rise to gametes. When reproduction occurs, the affected individual passes the defective DNA to the next generation. In somatic mutation, normal body cells change by chance and the DNA defect appears in every cell derived from the affected cell. Somatic mutation is evidence that none of us have a single permanent genotype. The genetic makeup of cells can change over time. Polygenetic inheritance. A particular trait results from the combination of multiple pairs of genes. Patterns of inheritance that either produce a trait or do not are easier to trace than polygenetic traits that vary along a continuum, such as height, intelligence, and personality. Polygenetic inheritance is complex, and behavioural genetic researchers often infer the influence of hereditary when precise patterns of inheritance are unknown. Harmful recessive alleles account for some developmental problems. Others are caused by chromosomal abnormalities. Most chromosomal defects occur during the process of meiosis. A chromosome pair fails to separate properly or part of the chromosome breaks off. The most common chromosomal disorder is Down syndrome. Down syndrome is produced by the presence of an extra chromosome on the 21st pair. It results in mental retardation, memory and speech problems, and slow motor development. It occurs in about 1 out of every 800 births, and the risk increases with maternal age. There are a number of disorders that arise from abnormalities in the sex chromosomes. Klinefelter's syndrome affects about 1 in every 400 males and involves the presence of an extra chromosome. Triple X syndrome involves the presence of an extra chromosome. So this is an experiment. I would love your feedback. Also, I am looking for podcasts that relate to what we are discussing in this subject. So let's all have a look, and maybe we can have a bit of a discussion of that in the discussion forum. So thank you all for listening. Some interesting kind of background on the science of development, I guess, and on chromosomes. So thank you, and I'm going to stop the